there is DNA sequencing. DNA sequencing is when you're looking at all the different nucleotides in the DNA. There was a human genome project. I think it was completed in 2001, and they sequenced 3.2 billion base pairs for the human genome. And after doing that, they completed that. After doing that, they found out of the 3.2 billion, only about 20,000 to 25,000 code for proteins. So only 1.5% of human genome encodes for protein. Then you might think, what is the rest of 98.5% doing? So if you remember, when we went over DNA replication, there was a step where RNA becomes a mature RNA. There were those intron regions. So there are a lot of intron regions. They don't code for anything and they get removed when it goes through transcription. So they don't know how that happens. They're still doing research. They have no idea how the body is doing that. Sometimes it can also code for regulatory proteins that kind of control other proteins, but only 1.5 is actually expressing or encoding for proteins. The cost to do this was around 100 million in 2001, and now it's 2021. Now you can do it for $1,000. So sequencing DNA, it can be useful for reducing protein sequences and to determine evolutionary relationships. When we say evolutionary relationships, what it means, now we have the human genes. We know, what percentage is encoding for proteins, what base pairs are there. You can do same thing for a cat. Then you can compare them and you can see how closely related humans are to cats or to chimpanzees maybe, or to monkeys. So you can take genome of different species and you can start comparing it. And we're gonna go over that when we go over evolution and natural selection. So it can be used to compare the species. Another thing that might be happening in the future, insurance companies might have everybody basically sequence their DNA at birth, and then depending on, they can look at the genes, and if it's coding for a genetic disorder, they can charge you high price. So another thing that can be done, they can look at the sequence and they can see which, for some of the diseases that they know the sequence for. If you have the sequence for that, then you might get the disease in the future and you might want to do something about it now. So that's something that can be done after you sequence your DNA. And they're also using it for criminal justice. So there was a case that was in news. I was some time ago. So there are these websites called like ancestry.org. They send you you swab your cheek cell and you send them your cheek cell, or I don't know, hair sample. They can sequence your DNA. So there was a serial killer and they couldn't find him for 10, 12 years. And then his granddaughter, I think, did one of those tests where she got her DNA sequenced and they were able to find a match and they were able to catch him like that. So, I just want to make sure you guys know, once you do this, government has your DNA sequence and they're going to have that information. Next technique we're going to talk about is called PCR. PCR stands for polymerase chain reaction. Polymerase chain reaction is where you are replicating DNA, but you're doing it in a test tube. Have you guys done the lab on Labster where you have to do PCR? Anyone? I think it takes you on a crime scene, get a hair sample, then you do the PCR. Not yet? Okay. So PCR is DNA replication. So let's say you pick up that hair sample from a crime scene, you only have one hair sample. You want to have enough DNA in order to identify or do anything with it. So you want to use PCR then. So to do PCR, obviously you need a test tube. And then if you think about DNA replication, Think about the enzyme that's needed for that. One is DNA polymerase. Then you have your A's and T's and G's and C's. Those are nucleotides. And then the primers are something that's going to tell the DNA where to start and where to stop adding the nucleotides. So in this test tube, you're going to put the DNA polymerase. You're going to put the nucleotides, your A's and T's and G's and C's. You're going to put the primers, usually 2. You can get these primers depending on the gene you're interested in. There are a lot of websites. You just go there and you say, I'm interested in this gene, and they'll design the primers for you, and you can just order them. And if you decide to do PCR, you can put them in here. So PCR, it rapidly produces millions of copies of a selected DNA sequence each time. So there is multiple cycles that you can do. Let's say each cycle is going to double the amount. So you start with the double-stranded DNA. After one cycle, you're going to have 2 copies. After 2 cycles, you're going to have 4. After 3, you're going to have 8. After 4, you're going to have 16. So it's going to double the amount each time. And it's going to give enough DNA for the investigators to analyze. So this is showing you that there is temperature increase that's happening, or temperature decrease that's happening. So I don't expect you guys to remember the temperatures, but just for your information, I'm gonna tell you that they increase the temperature at first. What is the first enzyme that's needed when you do DNA replication? You guys have to know 3 enzymes for that. What is the first one?
Helicase.
Helicase. Helicase opens the double-stranded DNA. In this case, you have your double-stranded DNA and you want to open that. The way that's done there is the temperature. Once temperature is high enough, it's going to be single-stranded. And now the primers can go attach wherever they need to attach, and they can start adding the nucleotides. And each time this happens, it's going to give you double amount. Another thing I want to remind you, if you guys remember, we learned about proteins, and we said, oh, protein denatures at high temperature. So these amino acids that can code for proteins are found here, so it can denature at high temperatures. So how do you keep this from denaturing? Because it denatures, it loses the structure, it loses the function, then there's no point of having this. So what they found, I think hot springs, they found this bacteria that can live on very, very high temperature. So from there, they were able to get the Taq polymerase. You don't have to know the name, but that's what it's called after that bacteria. It's called Taq polymerase. So that's the DNA polymerase. So they were able to add that to the sample. So once that's done, then it doesn't denature. So it can withstand high temperatures. So target strands are separated by heat. Then polymerases synthesize a new complementary strand. When I say complementary, again, it means A goes with T, C goes with G to match them. The number of molecules of DNA doubles with every round of PCR. I think hopefully you guys understand that. Let me also draw that out for you. So this is called exponential growth for PCR. So you start with your double-stranded DNA. After first cycle, what is the number that you have? Because it doubles. So you're gonna have 2. What about after second cycle?
4.
4. And after third one, it's going to have 8, double that. And after fourth one, 16. So I really want you to remember this. PCR doubles the number of doubles the DNA after each cycle. And you can put it for 30 cycles. That's going to give you billions of copies. And that can happen within 2 to 3 hours. Doesn't even take that long. So this test tube is put in a machine called PCR. So PCR separates DNA strands, uses enzymes to replicate each strand, and then repeats the process and creates millions of copies of the target DNA in minutes. So this can happen really, really quickly. I said 2 to 3 hours. That's for the machines that we have in the labs. They have more advanced ones that can do it even more quickly. Then it says the PCR is used in DNA profiling. So DNA profiling is when they are comparing DNA. So there's, since we are, humans is one species, there's not too much different in our DNA. So they can look at some regions that they know are different between people. That's DNA profiling. So they use PCR to amplify, means making a lot of copies of DNA from a tiny biological samples, such as those left behind at a crime scene. It can be, a hair sample. And PCR copies are highly variable target sequences throughout the genome. And then once they have enough DNA, they can detect genetic differences between between individuals. That's DNA profiling. So they can do that after they have enough DNA. There is a question for you guys. Suppose that you start with one double-stranded piece of DNA in a reaction tube. After 5 rounds of PCR, how many pieces of double-stranded DNA will be in the reaction tube?